Bonjour à tous, je vais vous présenter.
Good morning to all. I'm going to present the results of the AKER program from the genetics point of view. The first part of the AKER program aimed to identify genetic diversity, build up an allelic reference collection, and crossbreed with elite material in order to increase the usable genetic diversity and accelerate genetic progress. The second component consisted in an original way of making the most of all the diversity acquired, controlling it and evaluating it using new methods of phenotyping and high-throughput genotyping. The AKER program was therefore deployed in 3 stages. The first one consisted in identifying 16 reference planes from 10,000 genetic resources. The second involved crossing this reference collection with elite material to obtain source population, and the third step consisted in evaluating and analyzing these source populations, taking into account the year and the place effects. The great originality of AKER is to have made the hypothesis that there was a useful allelic variability in the unexploited germs plasm, and it was not necessary to make an a posteriori selection, but an a priori selection by genotyping before phenotyping. Previously, plants were evaluated in the fields and genetics were used. In AKER, we chose to start by using genetics and then evaluate the results of this selection. The program therefore aimed to go in search of existing diversity and then to incorporate, organize it and value it so it is always available to react and face new constraints. The allelic variability of the beet is essentially preserved through 2 international networks, Green ARS in the USA and Eurisco in Europe. AKER's first task was to list all the accessions available throughout the world and to try to collect as much information possible to describe them from a biological point of view, genera, species, subspecies, cultivars as fast as possible, and from an eco-geographical aspect, longitude, latitude, altitude, whatever. You should know that the beta genera is divided into 4 sections, as you can see on the diagrams on the screen. Only the species in the beta section cross easily with cultivated sugar beet, so this is a section we have favored in the choice of accessions. Geographically, the species of the beta sections are mainly found around the Mediterranean, around the Atlantic coast from Cape Verde to Denmark, and also in the Caucasian regions. The center of origin of the species will be probably in Asia Minor. From all this data, a list of 3,000 accessions has been drawn up, the distribution of which you can see here in terms of species and subspecies. This sampling was done by geographical representation, covering all the countries in which wild Beta maritima, Beta macrocarpa and Adanensis are found, and by biological representations. About 50% of the accessions selected were both from cultivated germplasm, all beet cultivars, but also fodder beet and vegetables. All these 3,000 seeds were ordered from the various gene banks and sown in order to be able to take DNA samples. The DNA collected, which was then used to first have a dark chip with 5,000 markers and then SMP chip with 30,000 markers of the sequences of the core selection. No less than 165 million pieces of data were generated and analyzed. Here are the results of the diversity analysis of the 3,000 accessions. On the first figure, the geographical distribution of the allelic wealth. As you can see, the lot of allelic richness is distributed everywhere, not only around the center of diversity. There are many hotspots, particularly in the French and English Atlantic coasts and around the Mediterranean. When we analyze this allelic wealth in terms of numbers, genetic distance, we can structure this variability. This is a structure that corresponds to botanical data. You can see the accessions of Macrocarpa or Adanensis species are very distant from the Beta maritima pool or cultivated pool. This is consistent with the known data. What's very interesting to note is that this observed diversity is also structured on geographical origin. You can see on the third graph the color gradient from northwest to southeast. This justifies fully the AKER's initial assumption was to select 3,000 accessions per crop group and per country, and this validates our approach. The compilation of all this data allowed us to choose with great robustness the 16 reference plants. Thanks to previous genetic analysis and the use of the AKER chip, we were able to show that 40 plants were sufficient to cover 100% of the total allelic variability. We were also able to demonstrate that 16 plants covered 100% of the complementary allelic variability of our elite material. These 16 plants constitute our core reference collection. All these plants constitute a representative sample of the total genetic wealth. You can see on the map that it covers a rather large geographical territory from Morocco, Denmark, through Israel, China, or the United States. It also covers all the species, subspecies of the beta genus: fodder, vegetables, sugar, Marittima adenensis, and Macrocarpa. The aim of the AKER genetic material creation is to pulverize the pre-exotic DNA carrying variability with successive crosses in the elite material. For this purpose, from 2013 onwards, the 16 reference plants were crossed with elite material to introduce new alleles of exotic origin by natural crossbreeding. In 2014-15, the F1 crossovers were successively backcrossed by the elite. The aim of the backcross was to increase the elite share while keeping pieces of the exotic plant. In each generation, 100,000 plants were produced with drastic legislation based on molecular data. In 2016, the plants reserved from crossbreeding were fixed by self-fertilization. And finally, in 2017, field hybridizations, 8 kilometers of cages, were carried out on the same female plant to represent the variability of the 3,200 male genotypes. You can see the spraying of the exotic fragments in blue or green in red. Each line corresponds to a plant, 200 hybrids per population, representing about 200 fragments spread over the whole genomes and overlapping each other, so no single piece is lost. In 2018-19, the hybrids obtained were phenotyped in the field in a multi-local evaluation system with experimental designs adapted to the size of the platforms. A number of evaluation criteria were selected so that the white sugar yield per hectare, root yield, and percentage of white sugar, resistance to seed set, resistance of foliar diseases, mainly oidium and cercospora, growth dynamics, root conservation, high rhizome pressure, etc. The white sugar yield was evaluated using 6 test platforms spread across the north and the Paris area in 2 rehearsals and 3 years of testing. A digger created for the AKER program enabled the weight of the roots to be measured directly in the field. For seed climbing, 2 observers were set up on the Côte d'Opale with sowing in mid-February and a count during the cycle of the number of climbs per plot. Tolerance to cercosporiose and oidium, the 2 main beet leaf diseases, which is a particular focus of attention in the context of reduction, suppression of phytosanitary products, was evaluated thanks to 2 observatories whose regime was carried out by multispectral imaging on board a drone. The extended duration of beet campaigns has an impact on the need for quality of conversion of the roots after uprooting and before processing in sugar factories. We have developed predictive methodology for this confidation quality through physical resistance analysis. This trait has been noted. The phenotypic version of the 3,200 hybrids was made possible through the implementation of multi-local, multi-year system with particular experimental designs in the fields. This is on a scale that has never been carried out before. 7-hectare plots, 120,000 plots in total, 10 experimental sites, 3,200 candidates with genetic commercial controls were added. 13 populations tested in 2018 and 2019, and 3 populations for which crossbreeding was more complicated were phenotyped in 2019 and 2020. Furthermore, the development and use of new statistical and analytical tools, aided by UAV phenotyping, have ensured that best results were highly robust. We gave ourselves the resources to obtain reliable results. A classic statistical analysis was carried out using different software on the 16 reference population made up of 200 genotypes. This was a special analysis of the average value of the sugar yield per hectare. At the same time, for diseases and seed settling, the progression curve of the area under disease, IUDPC, was calculated. It's a quantitative measure of the intensity of the disease over time. The data were compared to the genetic control, the elite used for backcrossing. The exotic fragment modulated the genetic expression of this elite and gave phenotypes that revealed these effects, whether positive, negative, or neutral, on the measured trait. In this way, useful, beneficial, or toxic genetic resources can be identified. At the same time, we also make comparison with commercial controls, the best varieties in the moment. The latter being the result of genetic progress achieved through traditional breeding, whereas for the AKER program, retrocrossings were made with elite or commercial material from 2012, the starting date for the program. The difference is therefore significant, 1% genetic progress per year on average. As you can see on the screen on the left, the distribution of root yield for the whole population. You can see this distribution is very spread out, with yield values varying from 80 to 112% of the controls. There is great interpopulation but also interpopulation variability. Some population performing better on average than others. On the graph on the right, the data, as an example, on the 6 fields population 8 or 3, to show the variation between fields but also within fields, and the differences in performance of the different hybrids. The second way, consider in the observation of distribution of phenotypic values of the individual of the 16 population compared to the genetic controls, the data represented by criterion. You can see here on the screen the data of 3 populations for sugar content, sugar yield and cercospore resistance. As you can see, there is genetic variability and therefore potential phenotypic potential. There are different forms of variability between population depending on the trait being measured. This shows that the potential for the sugar yield and cercospore resistance is higher than the potential for sugar content. The elite material used already showed a good sugar content close to the physiological threshold. You can see that for this criterion, the maxima are very slightly higher than the elites, and the average introduced physiologic resource rather regress the value of the correct sugar beets. Rare exotic fibers bring an improvement, but it remains low. On the other hand, the root yield is apparently still well below a ceiling. Variability is high, and we can observe individuals bringing high gains. The distribution is wide, demonstrating existence of a large number of genomic regions involved. A third type profile can be observed with a bimodal distribution. This is not to be the case for disease scores. These variations are indicative of exotic flavors with a strong defect and discontinuity in the expression of variability, demonstrating a character under the only a few genomic regions. When we look at the ranking of results by criterion, we discover that they belong to certain populations rather than others. For the performance criterion, the yield criteria, as you can see on the screen, 6 of the 16 populations observed demonstrate their ability to provide genotypes with significantly better performance. We have on this table the percent of hybrids per population beginning 10, between 5 and 10, and so on, up to the negative impact on yield. For another criterion, such as cercospora or odium, other populations have been highlighted. The most promising ones were all tested for 2 years, and the results show a consistency in performance of the main accessions chosen between 2 years of testing. The multi-year confirmation is all the more reason to support the robustness of the experiment in the face of the multi-year variation. The experimental system also made it possible to identify genotypes that are highly adaptable to a wide variety of environments. All these results give a first picture of usable variability, but it is of course important to look more closely to distinguish which one will be useful in the short and long terms. You have a zoom in on the best AKER hybrids. You can see 2 different profiles. A includes the AKER hybrids that are particularly high performing, with white sugar yields 5% higher than commercial controls, but sometimes show a few small defects in other criteria, low content sugar, poison to leaf diseases. On the other hand, the hybrids in profile B are a little less performing, 3 to 4% higher than commercial controls, but perform better on other criteria, particularly leaf diseases. It is therefore important to look at the distribution of this variability with individuals. At this stage, we don't construct new genotypes by successive crossbreeding, but the diversity of performance, whether in the same genotypes or not. For some individual, the potential is little less than what we could have had if we combined all the exotic fragments together. The most objectives of AKER's objectives was to compare phenotyping and genotyping data. We know the genomes of the 3200 hybrids perfectly well because we sequenced the genome of 16 founders and followed the introgress species with precision into a known and also sequenced elite genome. At the same time, with robust phenotyping data is available. So we can therefore try to highlight QTLs as the genetic association associated with the observed phenotypic variations. In this way, analysis can be carried at the level of chromosome traits, taking into account or not the data collected at the level of population. 1,400 areas were lighted out of 30 population tested in 18 and 19 for the criteria assessed. There remains to be analyzed 3 population tested in 2019-2020. QTLs were detected in all 9 bead chromosomes. The number varies from one population to another. Although some are found in different populations, QTLs are often specific to a population and therefore to the exotic reference accession, which therefore support the choice of the core collection. Only 14 QTLs were detected in 2 populations and only 1 in 3. For performance-related criteria, 320 QTL express a positive effect on the exotic allele, approximately 1/4. They are distributed on most of chromosomes, but more favorable areas can be distinguished, like chromosomes 7, 8, or 9. If we take into account non-yield criteria such as resistant to disease or seed set, 200 were detected and 89, so just under half, have a favorable effect. For diseases, fewer QTLs are detected, but they're largely favorable and therefore promising. They are detected mainly in 4 populations and appear to be common between diseases. For seed setting, many areas with negative effects have been identified should therefore improve the resistance of elite material, regardless of the annual, biannual aspect. We have to keep in mind that the 3,200 AKER lines correspond to a large reference collection, and if you want to study something new, we have to do it on all populations. Some populations are richer than others, some chromosomes too, but not always the same. If we look at the useful viability, we have introgressed roughly 2,600 exotic fragments taking into account overlaps, and I highlight 322 QTLs with favorable effects, about 12% of usable viability. This is the proportion I expect to find if tomorrow I study a new trait. By way of example, here I dissect the results for the root yield of population 803. A number of favorable effects are detected in green or unfavorable in red. For example, we highlight the genomic region on chromosome 3, for which a gain of 7% on average is obtained when the exotic alleles are present in the homozygous state. This is what you can see on the top right-hand corner. In this way, we could have in the supergenotype to have the variety of tomorrow. As a reminder, we have tried to summarize AKER's contribution to conventional selection in this diagram. Before AKER, working with genetics, a resource on which we had no information, was a laborious task, especially from exotic population whose yield level was extremely low. We first had to screen the material to identify a new resistance, for example, introduce the students into mapping populations to find the genomic regions of interest, and only then be able to follow this integration into new elite material from the backcross program. A long and laborious process, over 12 years before being able to submit a candidate to the CTPS, and not always rewarded because the phenotypic criteria measured on wild material are not always appraised once integrated into an elite and cultivated genetic background. Today, thanks to AKER, we have 3,200 lines well characterized both from phenotypic and genomic point of view, whose average years is that of the elite line used in 2012, therefore much better than a wild accession. We therefore have a huge reservoir of exotic fragments in elite background, directly usable because of a very acceptable level of yield, even excellent depending on the lineage. So we will therefore be able to easily draw from this reservoir to meet all the new challenges of the sector. We should therefore be able to submit candidates meeting this or that selection criterion in 7 years as opposed to 12 before. The genetic result of the AKER program brings significant advances on yield and richness of criteria, but also on disease and seed settling. These advances have been verified by QTL analysis, but the results obtained are a source of beauty for beet growing and for the sector as a whole. The AKER program will provide remarkable genetic material that irrigate future varieties for a long time to come. In the short term, we're able to detect a group of hybrids with confirmed performances that could be filed for registration at CTPS in 2022, the time it takes to multiply the number of parents. From then on, it will take another 3 years before it can be commercialized. In the medium and long terms, the AKER program will make it possible to produce high-performance varieties by introgressing and accumulating interesting fragments of the genome of reference plants in existing varieties. The AKER program thus offers a tremendous potential for diversity, those that are useful now and those that are unusual, even with a metabolic cost, but which could be useful later to answer new questions that will inevitably arise, such as resistance to certain bioaggressors like jaundice, or yellows, or water stress, and so on. It provides answers to questions that arise today, and will make it possible to answer the new questions that will arise tomorrow. Thank you to all the teams for taking part in this program, and thank you for your attention.